Hello, everyone. Today, our lecture is on statistical genetics. As you can imagine, with the discovery of DNA and the progress of computer technology, now we're able to analyze huge genetic data. Often, this accounts for terabytes of data. Lots and lots of information that can be processed, and nowadays people try to associate, scientists associate genes with various diseases, trying to find the remedies, treatments, and be able to predict certain features, traits for human population, as well as plants, animals. This is used a lot in agriculture for the selection purposes, to select the best products, fruits, vegetables, etc., that can be obtained genetically. But we have only one lecture on statistical genetics. We will talk about the very basic terms and basic foundations of statistical genetics. The foundations were laid by an Austrian biologist, Gregor Mendel, 19th century scientist. What Mendel understood is that the heritable features called traits are carried by genes. What are the genes? Genes are sequences of nucleotides that form the DNA. So genes are sequences of chemical substances, nucleotides, in our DNA. You have seen DNA. It's a long molecule that consists of many, many proteins. So the genes belong to the DNA. What else we need to know? We need to know that every gene comes in two forms. There are exceptions, but in the basics, the genes come in two forms: dominant and recessive. Dominant will be denoted by A, and recessive genes, little a. These are called alleles. So alleles are two forms of a gene. And therefore, you know that we humans have all the chromosomes in pairs, right? It's not always true for all living creatures. Some plants are multiploids, so we are diploids because chromosomes are in pairs. But there are triploids and multiploids, polyploids. In any case, we have pairs of chromosomes, and therefore we have pairs of genes. And since each gene can come in two different alleles, how many possible pairs of genes can we have? Each gene can have both genes in a pair in the dominant form, or one of each, or both in the recessive forms. The genes that appear in the same form, both capital, dominant, or both recessive, are called homozygotes. And mixed pair is a heterozygote. And each pair of genes, heterozygote, each pair of genes carries some feature, some trait. Often our characters depend on one gene, for example, colorblindness or hemophilia. Most features that we can see is a product of several genes, a combination of genes. For example, our IQ is often has an important inherited component. that comes from genes. So typically smart parents have smart kids, right? The color of our eyes, hair, typically depends on several genes. So it was noticed for millennia that kids, in a sense, are replicas of their parents. Blonde parents will likely have blonde kids. Tall parents will likely have tall kids. Of course, until Gregor Mendel, nobody knew why this is happening. And now we know that these characters, features are carried by genes. So they are passed from generations to the next generation, to the next generation, from parents to children with genes. So genes are passed from parents to children. Each parent passes one gene. So you have pair of genes, and only one of them is passed to the children. So every child receives one gene from the mother and one gene from the father. One gene is received from the mother and one gene from the father. Okay? And therefore, how can genes be passed? Let's see. Let's consider examples. So, let me connect. Okay, now I think I can write. Okay, so let's consider some examples. Suppose both father and mother have only dominant genes. So AA is the mother and as a dominant genes. AA is the father. What can be the child? Mother donates one of her genes. Father donates one of his genes. And as a result, the child will necessarily be dominant. So the child will have both dominant genes with probability 1. The child has no choice. Another example. Suppose both are heterozygotes. So the mother is AA, the father is AA. What will be the child? Well, now the mother can donate either the dominant or recessive gene, and the father can also pass dominant or a recessive gene. So the child can have any of the 3 combinations. What's the probability that the child will have both dominant genes? What do you think? Probability 1/2 of receiving dominant gene from the mother, probability 1/2 from the father. So overall, probability 1/4. The child can receive a dominant gene from the mother and recessive from the father, or vice versa. So this will have probability heterozygote, will have probability 1/4 plus 1/4, 1/2. The most likely combination. Therefore, if you compare the hair color, what do you think? You have more blonde people in the population, naturally blonde, legally blonde, the ones who don't dye their hair. naturally blonde people, naturally black people, or mixed brown. There should be more brown-haired people because that's a heterozygote. In fact, if you have a homozygote pair of genes, you will more likely inherit extreme features like blonde hair or black hair. When you have a heterozygote, it will be somewhere in between. Gregor Mendel studied the flowers of pea flowers, and found out that homozygote with both dominant genes will have red flower. Homozygotes with both recessive genes will have white flowers. And heterozygotes, what do you think, will have pink flowers, a mixture of red and white. And the child here will have both recessive genes with probability 1/4. Typically, various defects, unwanted features are seen with both genes being recessive. And there is a reason for that, the natural selection. If certain combination of genes is not favorable for living creatures, produces weak creatures, weak species that are vulnerable to diseases, then naturally this population will not propagate. So typically, if there are any unwanted features associated with the current gene, they're associated with both genes being in the recessive form. Okay, so we can see the split of probabilities is 1/4, 1/2, and 1/4 here. What else to know? So suppose you can see that the distribution of different pairs of genes, heterozygotes and homozygotes, may be different. So here, in this generation, the distribution was 1, 0, 0. With probability 1, we have both genes dominant. And in this population, it's 1/4, 1/2, and 1/4. So the distributions can be different. Let's fix the ideas, introduce some notation. So suppose in the population, the distribution of pairs of genes is U. That's the proportion of both genes being dominant. V is proportion of heterozygotes. And W is the proportion of recessive homozygotes. So these are three probabilities, and the sum of these probabilities, of course, should be 1, because there are only 3 possible combinations. Okay, then what are the proportions of dominant and recessive genes in the population? Then the proportions of dominant and recessive genes are, let's call them P and Q. So P, what proportion of creatures will have dominant genes? Rather, what proportion of all genes is in the dominant form, dominant alleles? Well, all creatures with both genes being dominant, so that is U, and half of the genes in the heterozygote population. So all of the U and half genes from heterozygotes. Similarly, what's the proportion of recessive genes in the population? Well, everybody with both genes being recessive is here, and a half of the genes from heterozygotes, the other half. So half of that. We need these probabilities to understand what happens in the next generation. So let's consider the next generation. We started with the population, the generation where the distribution of pairs of genes is U, V, and W, and the proportions of all dominant and recessive genes in the population in that generation is P and Q. And now let's pass to the next generation. Let's again consider examples. So example 1, suppose that in the population, let's say in this generation, all parents are heterozygotes. Suppose that all parents in this generation are heterozygotes. So what will be U, V, and W? U, how many parents will have both dominant genes? None. We assumed that all of them are heterozygotes, one dominant and one recessive genes. So this will be 0, none of them. Similarly, W is 0, and all the parents are capital A, little a, heterozygotes, so 1. What will be in the next generation? Their children. Well, we already calculated the population. If both the distribution, if both parents are heterozygotes, then their kids can be either dominant homozygotes or heterozygotes or recessive homozygotes. Therefore, in the next generation, U1 will be 1/4, V1 will be 1/2, and W1 will be 1/4 again. That's the new distribution, the distribution of genotypes. These pairs of genes are called genotypes. Distribution of genotypes in the next generation. So this is the distribution of genotypes in the next generation. Right? Okay, let's remember 1/4, 1/2, and 1/4 as we consider some other examples. So here, all the parents were heterozygotes. Okay, how about another possibility? Let's say all the parents are, so suppose the other extreme. So here, all parents were heterozygotes. Suppose we have homozygotes in equal proportions. Suppose parents are homozygotes in equal proportions. So U, proportion of dominant homozygotes, is 1/2, equal proportions. W, proportion of recessive homozygotes, is also 1/2. And we have no heterozygotes. So proportion of heterozygotes is 0. No heterozygotes. What will happen now? So every child has one parent. Doesn't matter, father or mother. AA. And another parent, AA, or AA and little AA, or both little AA. Right? These children will be AA. These children will be dominant, heterozygote, or recessive with probabilities 1/4, 1/2, 1/4. And these children will always be recessive. But how to calculate the proportions of all genotypes in the population of these children? Well, we can look at probabilities, proportions of dominant and recessive genes in the population. So we will have P, proportion of dominant genes. So that's U plus V over 2, right? U is 1/2 and V is 0. So that's 1/2. And Q, proportion of recessive genes, W plus V over 2 is also 1/2. Well, we knew that Q is 1 minus P. Q is 1 minus P, right? Because there are no other forms of genes. There are only two alleles, and one is the complement of the other. But, I mean, I would advise, just to check yourself, to calculate P and Q according to these formulas. Then you can check if the sum of them is 1. Just a double check. So, every child receives a dominant gene with probability 1/2 and a recessive gene with probability 1/2. Therefore, in the next generation, in the generation of children, the probabilities U1, probability of receiving both dominant genes, both father and mother giving the dominant gene. Each of them gives with probability 1/2, independently of each other. So far, the year 2025 is now. Father and mother cannot agree which genes they will pass to their children. And I guess that's good. Otherwise, it will be called genetic engineering. So the probability is 1/4. You multiply 1/2 by 1/2. Father and mother pass their genes independently of each other. W1, probability of receiving both recessive genes, is also 1/2 times 1/2, also 1/4. And V1, probability for a child to be heterozygote, is 1/2 times 1/2, one dominant, one recessive gene, or vice versa, recessive gene and dominant gene, right? Mother donates recessive or dominant, and then father donates the other form, the other allele. So probability 1/2. The distribution is 1/4 for the dominant homozygote, 1/2 for the heterozygote, 1/4 for the recessive homozygote. 1/4, 1/2, 1/4. Same as it was here. Also 1/4, 1/2, 1/4. Interesting? Let's try to, so we get, let's conclude. We get the same distribution of genotypes in the next generation. We started from different distributions, actually two extremes, but in one generation, we already converged to this distribution, the same distribution. Let's try to understand why this is so. Is it always the case? Let's try to consider the general case. General situation. General situation when we start with the distribution of genotypes U, V, and W. Sum of them, of course, equals 1. And then we calculate probabilities of genes. So proportion of dominant genes in the population, U plus V over 2, proportion of recessive genes, W plus V over 2, and then go to the next generation. So next generation. In the next generation, how can you become a dominant homozygote? Well, you should get the dominant gene from both parents. So the probability is p squared, right? You become heterozygote if you receive a dominant gene from your mother and recessive from your father, or vice versa. Recessive from the mother and dominant from the father. So that will be true PQ. And how can you be a recessive homozygote? Only if you receive both recessive genes from both parents. Each parent passes a recessive gene, multiplying, again, because parents do that independently of each other. So what will be the proportions of dominant and recessive genes among children in the next generation? So among children, that will be P1 and Q1. Proportion of dominant and recessive genes in the population of children. Well, we know the formulas. We already have the distribution of genotypes, and the proportion of dominant genes is U1 in that generation of children, plus half of V. Proportion of heterozygotes, recessive genes, is W1 plus half the other half of V1. Now we have formulas for U1 and V1. Why don't we use these formulas? So here is a formula for U, and here is a formula for V. Let's plug them in. What do we get? P squared for U, 2PQ divided by 2. That's PQ for V1, right? Well, P can be factored. P plus Q remains, but we know that P plus Q is 1, right? All the genes can have just two alleles, either dominant or recessive. So this is 1, so we get P. Look, P1 always equals P. The proportion of dominant genes does not change as we go from one generation to the next. And therefore, proportion of recessive genes also does not change. W1 is Q squared, and half of V1 is again PQ. So you can factor Q, and P plus Q is 1. So again, you start from Q1 and you end up with Q. So that's an important conclusion. We have P1 equals P and Q1 equals Q. This is called a stationary distribution. Stationary distribution. The distribution does not change when we go from one generation to the next. Stationary distribution. Okay. P and Q, the distributions of dominant and recessive genes, is constant. When we go from one generation to the next. when going to the next generation. And we just proved that we achieve the stationary distribution in one step. So no matter where we start, on the next step, we already achieve stationary distribution. So stationary distribution is achieved, the state converges to the stationary distribution in one generation. And this is a known result. This is called the Hardy-Weinberg law. Hardy-Weinberg law. Okay. If we were in class, I would now take a pause and ask for your questions. But since this is a remote lecture, if you have any questions, let me know by email. Meanwhile, we still have the practice sheet. So I published this sheet on Canvas in the Statistical Genetics module. Let's have some practice, shouldn't we? How about this question? Genotypes, dominant homozygote, heterozygote, and recessive homozygote, are distributed in the population with proportions 0.3, 0.6, 0.1. The sum is 1. This is a fine distribution of genotypes. Calculate the distribution of genotypes in the next generation, assuming random mating. What is random mating? Random mating means that we don't choose our mate, our partner, according to the genotype, dominant or recessive, homozygotes and heterozygotes, which may be true, maybe not. If you are blonde, will you seek a necessarily blonde partner? Probably not. Hair color doesn't matter. There are many more important things, right? When you seek your partner of your life. But if you're smart, probably you will seek a smart partner, right? At least in the human population. High IQ will probably want to mate with another high IQ. If you are athletic with strong muscles, will you seek an athletic partner? I'm not sure. Probably. So random mating is not a set in stone assumption. It may or may not happen. Let's see where random mating is important here. So how would you solve this problem? Well, based on the given distribution of genotypes, let's calculate the distribution of dominant and recessive genes in the population. So P is U plus half V. That's 0.3 plus half of 0.6. That's 0.6. And Q is W plus half V. And that's 0.1 plus the other half of 0.6. That's 0.4. Quick check that the sum is 1. Okay, we are on the right track. We calculated the distribution of dominant and recessive genes, and now the next generation genotypes. So U1, what's the proportion of dominant homozygotes? That's P squared, receiving dominant gene from both parents. So that will be 0.6 squared is 0.36. The proportion of heterozygotes we know is 2pq. So here I'm multiplying probabilities, right? Yes, I just told that fathers and mothers don't agree between themselves and they pass genes independently of each other, but they can choose their mates. That's their choice. So if people with high IQ choose other people with high IQ, then maybe the proportion of two dominant genes, one from the mother and one from the father, is more likely than proportion of heterozygotes. That's what we mean by random mating. Random mating means that the genes obtained from mother and father are independent. That's why we're multiplying here. So this is an important assumption of random mating, which is pretty much satisfied in agriculture when we're talking about plants. Or all the features, traits that you think are unimportant, not taken into account when humans choose their partners. So 2pq, 0.6 times 0.4 is 0.24, times 2 is 0.48. And W, what's the proportion of recessive homozygotes? That's Q square. 0.4 squared is 0.16. Let's check. 0.36 plus 0.48 is 0.84. 0.84 plus 0.16 is 1. Okay, that's just a check. So we were asked about the distribution of genotypes in the next generation. We found it. 0.36, 0.48, 0.16. That's the answer. That's the distribution of genotypes. Okay. How about the further generation of grandchildren in the same population? Well, how about we do the same thing? So in the population of children, proportion of dominant genes would be U1 plus half of V1. So 0.36 plus half of 0.48, that's 0.6. Q1 is W1 plus V1 over 2. W1 is 0.16. Plus half of 0.48, again, is 0.24. So that will be 0.4. The sum is 1, but also we notice that this distribution is the same as we had here, 0.6, 0.4. We knew that. That's the Hardy-Weinberg law. And therefore, in the population of grandchildren, U2 will be 0.36, V2 will be 0.48, and W2 will be 0.16 again. In the population of grandchildren, so 2 generations down the road, that's because we obtained the same P and Q as 1 generation ago. We converged to the stationary distribution. So this distribution of genotypes, it was here, and it is here again, does not change. That's the Hardy-Weinberg law. So we checked ourselves, we calculated the distribution of genotypes in different ways. When you are pressed in time, you can just copy the result from here, because according to Hardy-Weinberg law, we reach the stationary distribution in 1 generation. So going to grandchildren, it will not change. It will be the same distribution of genotypes. Okay, this is a nice theory by Gregor Mendel, and this all works with one exception, a crucially important exception. Now we will be talking about sex-linked characters. We're talking about gametes. Gametes are reproductive cells. Gametes. We are used to have the pairs of chromosomes. I just told that chromosomes come in pairs, and that's why we have pairs of genes. That's true. And you know that our body cells are multiplied. They multiply by just dividing. And each DNA is splitting, and therefore every cell, when it splits, it creates two copies, complete copies of its own. And therefore, all the cells in our body will carry the same genes. That's why now the science can take any part of our body and reproduce our DNA. And all the DNA tests in forensic science as evidences at criminal courts, all the DNAs will be evidences of our DNA. Doesn't matter what body part was used. You can submit your DNA test to find out your remote relatives on different sites, sites like familydna.com, heredity.com. There are so many different companies that can collect a sample of your DNA. You can send them a hair, or you can send them a piece of your saliva from the mouth. Does not matter, because the cells, the same genes, are in any part of our body. Gametes, the reproductive cells, are the exception, because that's where chromosomes are not paired. Females have the same two chromosomes in each pair, called XX. So they have X chromosomes, and their pair consists of two X chromosomes. Males, that's the genetic difference between females and males, have different chromosomes in each pair. I'm only talking about reproductive cells. So these are called XY. Then the mother, female, passes an X chromosome, X, to the child. Father can pass X or Y to the child. And this is roughly 50-50 chance. It may depend on the species. It's believed that there is a slightly higher chance to pass an Y chromosome. And if the father passes the Y chromosome, the child will be male or female? Well, the child will be XY, and that will be a male. If the father passes an X chromosome, the child will be female. So in that sense, the father decides whether the child will be a son or a daughter. So if the father preferred to have another gender of children, which I think is morally wrong, the father should not blame the mother for that. In any case, now we have a totally different story, right? The population of females have all three genotypes, AA, heterozygote, recessive homozygote, in proportions, again, U, V, W. So as we started before, but males have only one X gene. So the distribution of, they can't have a pair, only one X gene. So the distribution of dominant and recessive genes among females is, let's call it P prime and Q prime. Q prime. Okay, why do we talk about X chromosome? Most traits linked to sex are carried by X genes and not Y genes. And for this reason, if there is any specific feature, any disease, any unwanted feature, it's usually connected with X genes. And then the probability of this feature among males is higher. Examples of characters linked to sex are hemophilia. What is hemophilia? It's a disease where blood does not clot. So if a person with hemophilia has a cut, they start bleeding, and it's difficult to stop this bleeding. The blood doesn't clot. An example, the historical example is Russian Prince Alexei, often called Tsarevich. Alexei, it's a Russian word for a prince. He was tragically killed by Bolsheviks after Russian Revolution in 1917. The whole family of Russian Tsar was murdered in 1918. So it's known that Prince Alexei had hemophilia that he inherited. So who passed the gene with hemophilia to Tsarevich Alexei? That could not be his father, because the father passes a Y chromosome. So this proves that he obtained it from his mother, Alexandra, and it's now known for sure that this gene came from his very known, well-known great-grandmother. If you don't recognize, that's the English Queen Victoria. English Queen Victoria is a great-grandmother of Russian Prince Alexei. And if you've been to London and you saw Buckingham Palace, the main residence of the British royal family, they don't live here, but they live in Windsor, outside of London. But the main receptions, the main royal events happen here in the Buckingham Palace. There is a monument in front of the palace, and the statue here, that's Queen Victoria. That's the same queen. She had, this is proven now by DNA, she had the gene of hemophilia. So she passed this gene to Tsarevich Alexei. And as you understand, in the beginning of 20th century, more than 100 years ago, it was hard to stop bleeding when little prince started bleeding, and a person who knew how to stop bleeding, not completely known how, but he was able to stop bleeding every time for Tsarevich Alexei. That's Grigory Rasputin, a pretty controversial character, but you probably saw a completely untrue, not historically supported story of this family and Rasputin in the Disney classical movie Anastasia. In 1990s, Disney released Anastasia, a beautiful movie with a very nice story, and Rasputin is pictured as a monster there. A pretty controversial character, but at least he knew how to stop bleeding, and that actually made him close to the Russian royal family. Historical fact, so fathers cannot transmit any sex-linked feature to children. Most such features are associated with X genes, and X genes are always received from the mother. We can also conclude that these characters have probability P prime to appear in males, and probability, so to appear in females, they need to receive recessive X genes from both father and mother, right? So probability of a recessive gene, so no, Q prime, recessive gene, right? Well, we discussed this. Any unwanted characters are typically in a recessive gene. So probability Q prime to appear in males and probability Q times Q prime, so to appear in females, because they need to receive, to have the feature like hemophilia, they need to receive both recessive X genes from both parents. So the probability under random mating will be Q times Q prime, which is much smaller. Q times Q prime is much smaller than Q. Therefore, males are more prone to such diseases, these characters that are carried by sex-linked genes. In particular, probability of hemophilia, as I understand, is carried out by males, with the probability is about 1 in 4,000 males suffer from hemophilia, and for females, it's about 1 in 26,000. Very rare syndrome for females. Okay, so what else to know? So among females, the distribution of genotypes, we agreed, is UVW. The probabilities of dominant homozygote, heterozygote, and recessive homozygote. And probability of dominant gene is P, probability of recessive gene is Q. Among males, well, they only have one X gene, right? Not a pair, one X gene. The other gene in the pair is Y gene. So probability of dominant X genes is some P prime. Probability of recessive X gene is Q prime. right? And this may be different from P and Q in the population of females. Okay, now let's try to go to the next generation. What will happen in the next generation? Okay, let's now take our pen and calculate. So in the next generation, what will be the proportion of U1, the proportion of both genes dominant, V1, one of each, and Q1, both genes recessive. Okay, so to be dominant homozygote, you need to receive a dominant gene from the mother and a dominant gene from the father. Heterozygote, dominant from the mother, recessive from the father, or dominant from the father, recessive from the mother, right? And this is no longer 2pq because p is not necessarily the same as p prime, q is not q prime. Both recessive genes, that means recessive from the mother, recessive from the father. Okay, going to the proportion. So that's the proportion of genotypes, distribution of genotypes among children, next generation. Next, let's calculate the, sorry, that's not U1, that's W1, right? U1, V1, W1, the distribution of genotypes. Now, we will calculate proportions of dominant and recessive genes among children. And here we have to distinguish between daughters and sons. Daughters, so female children. They can have P1, Q1. So what will happen? And these also, by the way, when we talk about pairs of genes, we're also talking about female children. So that's about the distribution of genotypes among female children. We're talking about female children, right? So we know probability of dominant genes is U1 plus half of V1. So let's see, what will it be? Remember this U1 and V1. So we'll have P prime for U1, half of V1, PQ prime plus P prime Q from here, right? Over 2. Do you see how to simplify it? Let's try. I will make some room. Let's try to simplify. Bring to the common denominator first, right? So we will have twice P prime, P prime plus PQ prime, and another P prime plus P prime Q. I brought everything to the common denominator, so I have twice this here and here. I did it this way because now I can group the terms, and in the first group we'll factor what P, it's multiplied by P prime plus Q prime, which is 1. And in the second pair we can factor P prime, it's multiplied by P plus Q, which is also 1. So we'll have, since this is 1 and this is 1, we simply have P plus P prime over 2. And similarly, you can go through similar formulas. Q1 will be Q plus Q prime over 2. this is how the probabilities of dominant and recessive genes are calculated for the female population. For the females. How about males? Male children? Well, with males, everything is simple. They always receive X from the mother and Y from the father. So what's the proportion in the next generation, proportion of dominant genes? Well, it's just the probability of receiving a dominant gene from the mother. That's it. They receive X chromosome from the mother, X gene from the mother. So they receive a dominant gene with probability P, and they receive a recessive gene from the mother. They can only receive X from the mother with probability Q. Okay, and that's all they can get. They don't have a pair of X chromosomes. Okay, so we have different formulas. That's not Hardy-Weinberg law. Hardy-Weinberg law does not work for sex-linked characters. Or for reproductive cells. Okay, for the gametes. But there is convergence. So we don't get the stationary distribution. Let's make this conclusion. So we do not attain the stationary distribution after one generation. But the distribution converges. So we have, what do we have? We have what's called a difference equation in the distribution of females. Because with males, it's all very simple, right? They just inherit the probabilities of dominant and recessive genes from the female parent, from the mother. So in the distribution of females, the probability of dominant gene is calculated by averaging, right? Averaging the proportion of dominant genes in the mother and in the father population. So it will be the probability in the previous generation plus probability in the previous generation of males by 2. And what is this? That's, which I discussed, the proportion of dominant genes in fathers is proportion of dominant genes in their mothers, in grandmothers of children in generation N. So that's for the proportion of dominant genes, and similarly for the proportion of recessive genes. So to be recessive, homozygote, a daughter must receive a recessive gene from the mother and recessive gene from the father. And the father can only receive this gene from his mother. So it's a proportion of recessive gene passed from the mother to the daughter and from the grandmother to the to her son. These are called difference equations. As opposed to differential equations, these are difference, discrete version. See, they express Pn in terms of Pn minus 1 and Pn minus 2. I will not go through the mathematics. You had enough mathematics in my course, but you can just believe me that there is a general formula for the difference equations. I can derive it. If you're interested, let me know. There is a thing called characteristic polynomial. You can find the general solution of these equations, the general formula for Pn and Qn. What's important for us, this will converge pretty quickly. Not in one generation, but it will be close to the limit. And the limits will be 2p plus p prime over 3 for the proportion of dominant genes. Let's call it alpha. The limiting proportion of dominant genes in the female population. And Qn, 2q plus q prime over 3. Proportion, let's call it beta, limiting proportion of recessive genes for the female population, but these same proportions will be passed further to the next generation of males, right? The males have the same proportion with the lag of one generation. So when we talk about the limit, going one more step doesn't matter. Pn plus 1 will converge to the same limit. So this is for the entire population, for females and males. They will have same limiting proportions. Of dominant and recessive genes. Okay? So that is the story of sex-linked characters. Let's try to solve a problem. So this is the problem, number 4. Consider the gene located on an X chromosome. So that's X-linked character, X-linked gene. We have the same population, the same distribution of genotypes among females, 0.3, 0.6, 0.1. But among males, the proportions may be different now, 0.5 and 0.5. We have not reached the limit yet. Typically, in 3, 4, 5 generations, you get close to the limit. But now we have different proportions. Derive the distribution of all genotypes in the next generation. Let's try to derive. So what will happen in the next generation? Next generation. Okay, how about females? U1, proportion of dominant homozygotes. They need to receive dominant genes from the mother and from the father. So what is the proportion of dominant genes in the population of mothers? U plus half V, we already calculated it, 0.3 plus 0.3, 0.6, and Q is 0.4. The other half, 0.3 plus 0.1. We calculated in the previous problems. So here we'll have P, 0.6, times P prime, 0.5. That will be 0.3. V1, heterozygote. Daughters can receive heterozygote only when they receive different genes from the father and from the mother. So PQ prime plus P prime Q will have 0.6 times 0.5 plus p prime 0.5 times q 0.4. So it's what? 0.3 plus 0.2 is 0.5. So 0.3, 0.5, and W1. When you have a lack of time, it's the probability that makes the total equals 1. So it must be 0.2. But we have a few seconds. So probability of both recessive genes, if the daughter receives recessive genes from both parents, and q times q prime is 0.4 times 0.5. That's why it's 0.2. And 0.3 plus 0.5 plus 0.2 is 1. This is the distribution of genotypes, right? Distribution of genotypes among female children. How about male children? They only have P prime and Q prime because they don't have a pair for the X chromosome, and we're talking about a gene located on an X chromosome. So they only have one gene, either dominant or recessive, and they receive X chromosome from the mother. So this will be the probability P1 prime and Q1 prime, right? One generation further down the road. So they receive dominant gene with probability P. That's the proportion of dominant genes among mothers, so 0.6. And Q is proportion of recessive genes among mothers, 0.4. And this problem is solved. We have calculated the distribution of genotypes, and it's different among female children and male children. And the next topic that we will discuss with you is called selection. And selection refers to the intervention into the distribution of genotypes. This is used in agriculture when there are species, plants with unwanted characters, typically unwanted characters or traits are carried by recessive homozygotes. Typically, well, we discussed because of the laws of nature. If something is unwanted, then these traits will not propagate far through the generations. These are associated with more weaker, more vulnerable species. There are historical examples again. You know that royals typically marry royals. And that happened with the Spanish kings, the dynasty of Habsburgs, that mostly married within their large family. So any unwanted traits, any features were carried with their genes, and there was a high probability of receiving a gene carrying that character from both mother and father, if you're always made within a small group of people. The result? Well, the results are seen in portrait galleries. If you have seen the portrait, this is a king, Spanish King Charles II. You can look at the portrait and see that he's not well. He was a very sick person. Actually, this portrait is quite flattering. The artist tried to make it better than he was in reality. Maybe this portrait of the same king will tell you better. People who knew him wrote that it was difficult to watch. His mouth was not closing, fluids were dripping, and he died at a young age. Of course, people didn't know genetics at that time. They thought it's some piece of magic that was cursed him, and he got a nickname Bewitched. The King Charles II of Spain is known as Bewitched King. And also it's known that in the dynasty of Habsburgs, when you visit Spanish cities, Madrid has monuments to their kings at practically every plaza in Madrid historical downtown. Most of them wear this special collar. You saw what I'm talking about, because they used to have a very massive lower jaw. So their chins grew and grew through the generations. So to hide these chins, they invented these special collars. So that's also a selection process. They intentionally mated within the family, not knowing that this will aggravate their unwanted features. But in the selection process, in the agriculture, certain features may be unwanted. You don't want sour or bitter fruits. You want them sweet and juicy. So how can you intervene into the selection process? Well, simply to do the selection, the AA species are not allowed to mate. They will not be mating. How to do it? Well, you collect the pollen from the plants that you want to propagate through generations, and you don't include homozygote recessive genes in this pollination. Then you pollinate the field, and everything will be pollinated except AA species. How does this affect our calculations? So what will be the distribution of genotypes? So then the distribution of genotypes it was UVW, right? The proportions of 3 available genotypes, pairs of genes. Now, AA species are not mating, so it becomes a conditional distribution, right? We have a new condition that AA species don't mate. So it's a conditional distribution of mating genotypes. Only dominant homozygote and heterozygote are allowed to mate, right? So the distribution will be let's say U0 is the proportion of mating homozygotes, dominant homozygotes, is U divided by 1 minus W. So W is the proportion of recessive homozygotes, and they're not allowed to mate, so there will be no recessive homozygotes. That's the condition. We divide by the probability. Remember, conditional probability has probability of condition in the denominator. So we divide by the probability of the condition, probability of not allowing recessive homozygotes with probability W to mate. And similarly, V0, probability of proportion of mating heterozygotes, will also be divided by the same probability, 1 minus W. W0, that's probability of mating recessive genotypes. What is that? They're not allowed to mate. So 0, and that also makes the sum equal 1, right? It would be W, but it would be 1 if we put W here, but we eliminated W, so it became 1 minus W. Now we divide it by 1 minus W to make the sum 1 again. So we have a conditional distribution of mating homozygotes. Then the proportions of dominant and recessive genes in this distribution are P0 and Q0. So proportion of mating dominant genes that are passed to the next generation. So that will be U plus V over 2 again, divided by the probability of condition. AA doesn't mate. And probability of proportion of recessive genes, V over 2, divided by 1 minus W, and proceeding to the next generation. In the next generation, what do we have? UN plus 1, proportion of dominant genotypes, VN plus 1, proportion of heterozygotes, and WN plus 1 will not be allowed to mate, but it's present in the population. Recessive genotype. So what will it be? Well, you have to receive dominant genes from both parents, or one from each, or recessive from both parents. Right? So with that in mind, what will be now the proportion of dominant and recessive genes? We'll derive a formula. It will be, again, a difference equation, but luckily this will be a simple formula. We will be able to solve the difference equation and get the general formula. And how is this applied? Well, it's applied very directly in agriculture, in the selection process. If you are having bitter watermelons, how many generations will it take to get the proportion of bitter watermelons to less than 1%? That we can calculate by the formula. Okay, let's work a little. So the proportion of dominant genes in the next generation, U plus V over 2, so UN plus 1 plus half of VN plus 1 over 1 minus W, because they are not mating, WN plus 1. We can take the probabilities from here. So we have PN square plus half of VN plus 1, that's PN QN, over 1 minus WN plus 1, that's QN square. Let's simplify. How do we simplify? This is, we did similar things, right? Pn is factored, Pn plus Qn is 1, divided by 1 minus Qn square. 1 minus Qn square, you know, is 1 minus Qn times 1 plus Qn. You know this factorization, right? Why do we do it? Because 1 minus Qn is the same as Pn. That's the same thing, they're complements of each other. So the whole numerator becomes just 1, and as a result, we have 1 over 1 plus Qn. We simplify it. That's the proportion of dominant genes in the next generation. Let's calculate Qn plus 1, the proportion of recessive genes in the next generation. So the proportion of recessive genes is how can you receive recessive gene? W doesn't play the role here. The recessive genes don't pass, the recessive genotype doesn't mate. So you can only receive recessive gene from a heterozygote divided by the same condition. So that becomes half of 2 PNQN is just PNQN. Now it's correct. Divided by 1 minus QN square. So we factor the same way. 1 minus QN times 1 plus QN. That becomes, so PN is the same as 1 minus QN, QN over 1 plus QN. And that's great because now we have a recursive relationship. We have a recursion. 1 over QN plus 1 appears to be the same as 1 over this, 1 plus QN over QN. And that's, let's divide term by term, 1 over QN plus 1. Excellent. So the reciprocal of QN has a nice recursive relationship. The reciprocal is always incremented by 1 with every next generation. So therefore, we can have a general formula, 1 over QN is 1 over the initial proportion of recessive genes. Every generation adds 1. So n generations add n. That's the general formula. And therefore, if we want the proportion of recessive genes, so take a reciprocal of that. 1 over n plus 1 over q0. That's the proportion of recessive genes. Proportion of recessive genotypes is qn squared. So 1 over n plus 1 over q0 square. That's the proportion of recessive genotypes. And now, if you want this proportion to be less than a certain number, then you can solve this equation with respect to n. And that's what we'll do in this problem number 3 in our practice. In the same population, so in the same population as we had here with genotype distribution 0.3, 0.6, and 0.1. Okay. In this genotype distribution, so the probability of recessive genotype was 0.1. It's too much. You want to reduce it from 0.1 to 0.01, from 10% to 1%. How many generations will it take? How would you answer that question? Okay. We can calculate how many generations it will take. And for that, we need to calculate Q0, right? So Q0 is V over 2. Normally, we would add W, but W does not propagate the recessive genotype. Divided by 1 minus W. So V was 0.6, right? The distribution was 0.3, 0.6, 0.1. So half of 0.6 divided by 1 minus recessive genotype had probability 0.1. I'm taking it from this initial distribution. It says in the same population, so same distribution. So Q0 is found. It's 0.3 divided by 0.9 is 1/3. Then Wn is we just derived 1 over n plus 1 over q0 squared. So that will be, but that's in generation in the previous generation, n minus 1. Wn is qn minus 1 squared. You receive the two recessive genes from the parents. So n minus 1 squared. So n minus 1 here and here. N minus 1. You receive two recessive genes from the previous generation, from parents. And therefore, it's 1 over n minus 1 plus 3, reciprocal of 1/3. Minus 1 plus 3 is 2. Okay? The problem wants Wn to be 1%. So equate and solve it. When 1 over n plus 2 square is 0.01, you can solve it. That means 1 over n plus 2 is 0.1. That means n plus 2 is 10, right? And that means n is 8, right? So we need 8 generations to reduce the proportion of unwanted recessive genotypes from 10% to 1%. 8 generations. All right, that concludes our lecture. I want to show that this material that we just discussed is in our lecture notes, chapter 12. There is another section that has a few exercises. If you're interested, you can solve this type of problems. If a dog has black ear, what's the probability that its grandson also has a black ear? What's the probability that the dog's nephew has a black ear? And if the probability is higher, that will tell you that they're closer relatives. That in that way, we're defining the degree of relationships. Which relatives are closer in their relationships? Well, the relatives whose probability of passing genes is higher. So these exercises will shed light on how to calculate these probabilities. For example, passing genes from grandparents to grandchildren. Is this a higher probability than between aunt and nephew? So in that way, we are solving the degree of relationship problems. Of course, there are many other problems connected to statistical genetics. That concludes our lecture. Let me know if you'll have any questions.